So thank you to the organisers for inviting me to speak today. And today I'm going to talk to you about the differences in daily carbohydrate metabolism in Arabidopsis, Brachypodium, and in wheat. So in Arabidopsis, which I'm sure you're all aware is the model species often studied, we understand this best. And during the day, plants photosynthesize, and they fix carbon and use the sucrose that's produced for various growth processes and for general metabolism. But also during the day, they partition some of the fixed carbon into transitory starch. And then they use this starch at nighttime when they can no longer photosynthesize. And this starch is broken down into sucrose, which can then be transported all around the plant and used for growth at night. So why is this important? Well, actually, nighttime metabolism is incredibly important for the plant productivity. So this is just one small example. You can see on the left a mutant plant, which is a mutant in glucan-water dikinase. But the important thing is that this mutant can't break down its starch at nighttime. And as you can see, just from the size of the plant, it really can't grow as well as a wild-type plant. So this is just one piece of evidence that nighttime metabolism is really important for plant productivity. So in Arabidopsis, during the day, starch is accumulated. And then at nighttime, which is the part in grey, starch is degraded at approximately a linear rate. And then it nearly runs out entirely of starch by dawn. And this degradation of starch at nighttime is carefully controlled and has various inputs such as circadian control. So we know quite a lot about the control of starch degradation at nighttime in Arabidopsis, but we want to also know about other species. And in particular, we're interested in looking into wheat. So previous reports have stated that wheat accumulates sucrose instead of starch during the day. And then this sucrose is degraded at nighttime. We wanted to look into whether starch also accumulates sucrose in our conditions, which are controlled environment rooms. And also, we wanted to try and use Brachypodium, which is a model for cereals, to investigate this problem. And this is because Brachypodium has many advantages experimentally over wheat, because it has a much shorter lifespan, it's a diploid, and it has many more genetic resources available that we could use to study this particular metabolism. So first of all, we looked into Brachypodium. So as you can see, we compared the accumulation of sucrose and starch. And at the end of the day, Brachypodium has accumulated a lot more starch than it has accumulated sucrose. And this was quite a surprise, because as I said before, wheat has been reported to accumulate sucrose, and we expected Brachypodium would be more similar to wheat. But actually, it accumulates starch more. And then by the end of the night, this starch has been degraded so that nearly none is left. So we looked further into the pattern of starch accumulation and degradation in Brachypodium. And as you can see here, Brachypodium during the day accumulates starch, and then at nighttime it degrades it at nearly a linear rate until dawn, when nearly none is left. And this might sound quite familiar. It's because it's pretty much exactly the same pattern that's seen in Arabidopsis. And this was quite surprising. So we wanted to continue on, and really we wanted to focus on wheat. So we moved on to wheat because it appeared that Brachypodium wasn't really a good model for wheat in this particular case. So looking at wheat, we looked at the starch content of wheat over 24 hours as well. And as you can see here, the amount of starch accumulated in wheat is much lower than in Brachypodium and in Arabidopsis. And then we looked into the accumulation of sucrose, which is what was previously reported. And indeed, sucrose is accumulated during the day, and then it's broken down at nighttime. But you can see that actually the pattern of degradation is quite different, because the rate of sucrose degradation in wheat seems to be pretty rapid at the start of the night and then slow down, whereas in Brachypodium, the rate of degradation is approximately linear for the whole of the night. And this could have important functional consequences. So I've said before that Brachypodium accumulates starch during the day and degrades it at nighttime, whereas wheat accumulates sucrose and degrades it at nighttime, which is surprising since they are quite closely evolutionarily related. And we expected to see more of a difference between Brachypodium and Arabidopsis, than between Brachypodium and wheat. So we came up with two hypotheses for why this might be the case. The first of these was that wheat is obviously a domesticated crop, and maybe during the course of domestication, it's been selected for wheat to accumulate sucrose in its leaves rather than starch. And our second hypothesis was that maybe it's something to do with the ploidy level of these species. So wheat is a hexaploid, whereas Brachypodium is a diploid, and maybe this is the cause of the difference we observed. So to investigate this, we used the evolutionary history of wheat. So wheat is a product of several hybridization events between wild grasses. And the first of these occurred between two wild grasses known as wild einkorn and Arabidopsis beltoides. And this hybridization event caused the production of wild emmer, which is a tetraploid. This tetraploid then underwent another hybridization event with Arabidopsis tauschii to produce a hexaploid, which was domesticated into domesticated bread wheat. And this is the species which we've been working on in all the data you've already seen. So you've actually seen this graph before in a different form. So by the end of the day, which is the grey bar, sucrose is accumulated to quite a high level. But then during the night, the sucrose is degraded until nearly zero is left at the end of the night. So we wanted to look in these other species to see if a similar pattern of sucrose accumulation is observed. So looking back to the tetraploid ancestor, then you can see that also here, sucrose is accumulated and degraded during the night. A similar pattern is also seen in a diploid ancestor, Arabidopsis tauschii, which is a D genome donor. We then thought maybe this difference, that the first hybridization event between Triticum maratu and Arabidopsis beltoides might explain whether sucrose is also turned over in these species. But actually, both of these species also accumulate sucrose and break that down during the night. So it appears that domestication doesn't have an effect on whether a plant accumulates sucrose or starch, because all of these wild and domesticated species use sucrose. And equally, the ploidy level, where we have three different levels of ploidy here, diploids, tetraploids, and hexaploids, all use sucrose. So during this work, we've shown that Arabidopsis accumulates starch and uses it as its source of carbon during the night. And surprisingly, also, so did Brachypodium, which is very different from what we've seen in wheat, which accumulates sucrose during the course of the day and degrades that at night. And it'll be interesting to investigate the evolutionary history of this trait and see if it's been lost and gained multiple times, or if there's more of a broad pattern between species. And another thing it'll be interesting to investigate is what's this functional significance of using a different carbohydrate source during the night. And I'd like to end by thanking Alison Smith, my supervisor, and all the members of her group, and also Olawase Shoronola and Anya Kowalski, who did all the work on the wheat and the wild relatives.
That's very nice, Philippa. I'm supposed to say my name, Alex Webb, Cambridge. So do we know anything else other than wheat which accumulates sucrose, or is it that Arabidopsis and Brachypodium are the oddities?
So we actually, it's quite difficult to compare between different experiments that have been done previously, because obviously using different conditions, whereas for our experiments we use controlled environments, then it's more comparable between species. But we do know that barley accumulates sucrose as well. But maize, for example, accumulates starch. Rice accumulates both sucrose and some starch. So it's sort of a mixture between different species, but at the moment we don't really have definitive data to say whether, exactly where these different traits might have arisen in evolution.
In Arabidopsis, Matthew Hanna from Bayer Crop Science in Ghent. In Arabidopsis, for sucrose, is that completely flat across the day, or is that also raising during the day and there's a reduction during the night?
I think there's a very slight increase, but the order of turnover is so much smaller than starch that starch has been much more studied.
And are you looking just at these specific metabolites, or are you looking at other metabolites such as, I think there was a paper speculating on fumarate as a potential carbon store in some leaves? So have you been doing full metabolite profiling, or just...
No, we've just been studying starch and sucrose.
Thank you.